All right. Welcome back, everyone. Hopefully you're prepared to be entertained and educated. These two need no introduction. I'm going to welcome Dr. Ed Wild and Dr. Jeff Carroll to the stage so they can inform us which wolf will win.
Cheers, man. Hi, everyone.
That was loud.
How are we doing? Everybody made it to the bathroom, 7 miles away.
So I want to set the stage by letting you guys know that we just found out we're not going to be able to see the next slide. So I have absolutely no idea how this is going to go. And I literally have no idea what the title of the talk means. So we're starting off an early.
But the great news is on the screen in front of us, we can see video footage of ourselves.
Oh, that's nice.
And not the slides. But
Oh,
we'll
it's
get there.
It's slightly
We'll get
delayed,
there.
so it's a little strange.
Some people don't know who we are. I'm Ed. I'm the English one, which is why I stand on this side, because Europe, America, because otherwise you'd have no idea which is which. This is my real voice. I speak like this the whole time. I dream in this accent. Oh, and I'm a neurologist and an HD researcher.
My name is Jeff Carroll. I am a Huntington's disease researcher. I run a lab. I study Huntington's disease in mouse models. I'm also an HD family member. My mom died of Huntington's disease about 10 years ago, and a member of a large family affected by Huntington's.
And the title of our talk is Which Wolf Wins? Because in about February, George emails me and says, what are you going to call your talk? And whatever film I watched the previous day informs my philosophy for the next 24 hours. And the film I was watching was Tomorrowland. So if you've seen that film, you'll know which wolf wins, but don't spoil it for everyone else because it comes right at the end. So it's 25 years and I think 74 days since this paper was published in the journal Cell. This was the announcement that the Huntington's disease gene had been discovered after a huge effort from a ton of researchers and, more importantly, HD families. And so this is the 25th anniversary, the silver anniversary of the discovery of the Huntington's disease gene. So it's a significant anniversary. And there's this saying, every cloud has a silver lining. And I'm sure most or all of you were here for the previous session in which we heard some pretty cool stuff and news of a significant breakthrough. It's made our job really difficult because normally we stand here in our research roundup and talk about all the cool stuff that's happened over the past year that you probably haven't heard about before. But the main thing we were going to talk about if that was our remit would be this trial. What we want to do is briefly revisit that trial, what it means and what's next, and the bigger picture, and give you guys kind of an action plan for making all of this work together. And then we're going to zoom out into the even bigger picture of some things that are much earlier in the pipeline but could produce cool news in the future. And this is how we started when we first gave this talk at Las Vegas in maybe 2011. It was basically, hey, there's this really cool stuff that's happening in mice called huntingtin-lowering or gene silencing. And then 7-8 years later, here we are at the meeting where the results of that trial were announced. And so what we want to do today is plant some seeds of some cool stuff that's early in the pipeline that could turn cool in the near future. So that's the plan. Cool? Okay. So you've seen this little 3D potato before. This is the Huntingtin protein, and as we heard earlier from Jim and a couple of the other speakers, the DNA Huntington gene is a recipe for that protein. And in Huntington's disease, too many CAGs in the Huntington gene leads to too many glutamines, these little pink blobs in the protein, and that causes the protein to become harmful. It's called the mutant Huntington protein. The word mutation it's from a Latin word, and it simply means changed. So it has some associations, but it's not fundamentally a word that means something's good or bad. It just means it's changed.
Nancy Wexler suggested to me yesterday that we should start calling it the fancy version of the gene, which I really like. So thank you, Nancy.
Yeah,
I think
it has a little extra something. And this is what it looks like. It builds up in cells, and it looks like this blob, this mountain. And the stuff that's probably the most harmful is kind of floating around in this soup at the foot of the mountain. And that causes problems. It interferes with many things in our cells. It's the reason our brain cells start to malfunction and eventually can't cope anymore, and they die prematurely. And that's the fundamental problem of Huntington's disease. This is a rat cell in which you can see the blobs of Huntington protein building up, these bright green blobs. It's dramatically speeded up. It doesn't happen anything nearly this fast in humans. But we can see Huntington's disease processes happening under the microscope. And that presents us with, technically, theoretically, a pretty straightforward problem. We know the cause of our disease, and that's not true for Parkinson's or Alzheimer's. And over the past 20 years, scientists have developed tools for interacting directly with cells in a way that could switch off this problem at its source. And that's what we heard this morning. Jeff is going to very briefly summarize what we saw, but it starts with this basic dogma of biology.
So this is the central dogma of molecular biology. This is as much biology as if you're, say, a neurologist. This is how much you have to know. Our genetic information that we inherit from our parents, right, is kept in our DNA. And as Anne said this morning, it's a really beautiful way to think about it. The DNA is really important, right? So our cells keep it in the nucleus, in the central part of our cell, and they take care of it. But the genetic information encoded in our DNA is necessary for our cells to build proteins, right? The Huntington is this floppy pink potato thing, but it's a protein that does stuff in cells. And with the Huntington protein, we don't actually know what all those roles are, but we know they're important. And so when a cell needs a protein, it goes into its store of DNA, and it makes a copy of the specific genetic information it needs for whatever protein it's after. And it makes a copy in a chemical language that's related to DNA. It's called RNA. And because this is a message carrying information from the genes to the protein factories, this is called messenger RNA or mRNA. And oh, do you want to talk about?
Right. So when we first started giving these talks, you will have heard us call it gene silencing, and that was what everyone called it. And it's quite a cool name, but there's concerns that might suggest that this gene is going to be turned off completely, rubbed out, obliterated, no protein is going to be made. And actually there's reasonably good evidence that that's a bad idea. Getting rid of this protein completely could cause more harm than good. But the reality is, even if we wanted to do that, which we don't, we wouldn't be able to, because as Anne Smith said earlier on, the most we can achieve at the very highest doses is something like 80% reduction of that protein. So this idea of gene silencing, it's cool and catchy, and you'll probably still hear it, but the preferred term is huntingtin-lowering as it applies to Huntington's disease. So those are exactly the same thing. Nothing's changed about the science of the technology. It's just a sort of linguistic preference to emphasize that we are lowering the Huntington protein in a controllable way to a desired amount and not obliterating it altogether.
You may want a slightly less ominous photo for your Huntington lowering. That was a little more creepy. Okay, so I hate to sort of recap after that beautiful introduction we had this morning, but there's folks watching at home, and so I want to go over the highlight that we heard in a little bit of detail, and talk about this drug that was developed by Ionis Pharmaceuticals, this so-called antisense oligonucleotide. What's unique about these kinds of drugs is that they're not little small chemicals. They're actually drugs made out of DNA. In fact, heavily modified bits of DNA. That's a single-stranded bit of DNA that scientists at Ionis and other places have worked for many years to sort of chemically hack so that this DNA bit can get into cells and, as we heard today, even travel throughout the brain.
And actually, sorry to interject, but Blair showed the photo of us visiting Ionis to see the results of the trial. We also had a tour of the Ionis laboratory, and I've been saying to audiences like this for like 10 years, guys, there's this really cool machine in California that makes DNA. I saw the machine. It was such a geek out. There's photos. We interrupted a whole afternoon's work. There's a machine, and you type the DNA sequence, and there's 4 bottles connected to it, and they're labeled A, C, G, and T. It's crazy. It's exactly like how you dream it's going to be.
HD Science shut down for a day so Ed could take a photo.
It was so cool.
So the way that these Huntington lowering techniques, and there's a lot of them now, and we'll keep talking about the different approaches, they all have the same idea, which is reducing the levels of the Huntington protein, because it's levels of the Huntington protein, we think, not the DNA or the RNA, that are harmful to cells. So the goal of all of these therapies is to, in one way or another, interfere with this process by which genetic information gets translated into proteins, with an idea of reducing the Huntington protein. in the case of Ionis and other companies that work on these so-called ASO drugs, the single-stranded bit of DNA comes into the cell. It finds the one gene that it matches, in this case the Huntington gene. It ignores the like 24,000 or whatever other mRNAs that are in the cell, and it causes the degradation of that mRNA. So our cells have this degradation procedure that breaks down structures like this, and so when the ASO goes in the cell, finds its partner, sticks to it, the cell degrades that RNA. And so you've tricked the cell into destroying one message. And in our case, of course, that message is the message for the Huntington protein. And if you degrade the mRNA, you reduce the levels of the Huntington protein.
So we talked earlier on about how this drug has to be delivered, and to recap, you can't give it into an injection in the blood or a pill. It has to be injected directly into the spinal fluid. But as we heard earlier on from the trial results, that turns out to be really well tolerated. It sounds way worse than it is, and the reality is it's generally fine.
I have had a lumbar puncture. It wasn't like the best day of my life, but it was way far from the worst day of my life. That was even with Dr. Levitt doing it, so believe it or not, I let that guy do it. So it was a fun day at work. The trial that we're going to be talking about was the safety study run by Ionis to see whether these ASOs that target the Huntington protein for lowering. The first trial of any new investigational drug, but especially one that does something this cool and this advanced, the first trial is to look at safety. And specifically, as was talked about this morning, this first safety study was done in Huntington's patients, and it was an ascending dose study, right? So the idea is you give people a tiny dose, and then you wait and you watch them really carefully, and then you move up to the next dose. So formally, this trial was called the Ionis-HTT-RX trial. It was a phase 1/2A for the aficionados in the audience. It had several dozen patients around the world. There were not sites in the US. Sometimes people say, like, why not? Because these sites that did enroll these patients were the first ones that were able to do it. So remember, like, as an HD community, it doesn't matter to me where the phase 1 study is run, right? HD is HD around the world. So these sites were the fastest way that Ionis could get this drug done to test for safety. So the sites were in UK, Canada, and Germany. There was a placebo arm. You know, taking part in a trial this exciting makes people feel good. And when people feel good, if you ask them how their HD is going, right, they say, lots better, because I feel really motivated and good. So a placebo arm, although it's frustrating to think about, you know, being in this trial and getting a dummy drug, it's absolutely required to control for that kind of effect of how exciting it is to participate in this trial. I have no idea who this woman is. I had to put this in.
This isn't me telling Jeff to hurry up, although it'll do for that.
Who is this lady? Okay, apparently...
Jeff lives in an underground bunker with no internet access or connection with popular culture whatsoever.
It's called academia. I haven't had a real job my entire adult life. It's amazing. So it felt like
She's trying to tell us something, Jeff.
Been waiting for a long time for these results. And we finally got them last December, on December 11th, 2017, when the so-called top-line announcements of this Ionis-HTT-RX trial. This made headlines around the world, although as an HD Buzz guy, I have to critique some of these headlines, but that's another talk for a different day. The
press release was very cautiously written.
No, that doesn't surprise me.
This happened while I was on holiday in Hungary. I was on a long weekend in Budapest, and I spent the whole weekend on the phone to journalists, and my partner was not happy. Anyway, oh right.
Oh, I'm talking about those results. Ed can't talk about these because he did it, yeah. So these are the top line results. So just for folks in the audience who haven't thought about this before, sometimes it's frustrating, you want all the data from the study, but think about, like, we're measuring all these safety parameters, all these brain images, all these psychological tests. Now think about how many different ways there are to look at that data, right? Like this versus that. Did people who lost weight have better cognitive? I mean, there's a million ways to look at this data, and it takes time to do it carefully. So what Ionis has appropriately done here is they gave us a very careful and concrete answer to our primary questions for this study. That was what we found out in December. There were no serious adverse events reported in participants, and no participants discontinued from the study. This is an amazing result, and especially with a drug this interventional, right? I mean, spinal injections are no small thing, and you would think that somebody, especially a HD family, would, you know, eventually maybe tire of this and drop out, and not one single participant did, which is remarkable. And then the other, and I think most striking thing that I've seen since I started in HD science was a so-called dose-dependent reductions. And what that means is that this was a study in which increasing doses of the drug were given, right? That was part of the safety study. And if your drug works, then you should be able to say, if I give more of it, I'll get more Huntingtin lowering. And in fact, that's what they found. So this graph, as I've sort of described it to a couple people, was like the second number that changed my life. The first was when I underwent predictive testing in Vancouver in 2004 and learned that I had inherited my mother's mutation. I had a CAG of 42. And at that time, I thought, that's it. I'm going to die of Huntington's disease. I'll do what I can in the time between now and then. And that changed my life a lot. This graph was the next number that had that big of an impact on my life. Somehow, all the people I work with who work in the clinical trial kept it a secret from me. They really did.
That was really difficult.
So we were sitting next to each other in the front row when this slide was presented at a recent CHI conference in February, and it just, everyone teared up, right? It's just this amazing proof that this drug, which has been this incredible, arduous, beautiful process of science that you guys heard about this morning, does what it's supposed to do. It's just a remarkable feat.
And what's more, it's not just all or nothing. We have this dose dependence. So the bigger the dose of drug, the lower the protein goes. So we have a volume control knob for the Huntington protein. The meeting Jeff was talking about was this meeting a couple of hours away in Palm Springs, which is the CHDI therapeutics convention. And this was memorable for the reasons that Jeff has mentioned. Obviously, it was a packed house. They knew they were going to hear the first results of the trial ever presented publicly. They knew that they were going to hear it from Sarah Tabrizi on the right and Anne Smith on the left. Sarah, as I said, was the global lead for the trial. For me, it ended up being memorable, equally memorable for another reason, which was that Sarah, the joint talk was scheduled to start at a certain time, but the session was actually running, which is unusual, about 5 minutes early. So when Sarah and Anne were announced onto the stage, Anne got up on the stage, but Sarah was nowhere to be seen because she had gone to the lavatory. And I'm now proud to exclusively present a genuine screenshot from my phone. And she came back. Everybody knew where she'd been, and it was a nice way of kind of easing some of the suspense anyway, so that happened. The next update was a rather larger and more kind of formal occasion. This is Sarah's presenting the results at the American Academy of Neurology meeting in LA, actually, just downtown LA in April. About 8,000 people in that audience, and that was where we first heard the clinical results from the trial, which Blair presented earlier, that relationship between the amount of lowering of protein and the change in the clinical scores, which, as we've said, is not evidence that the drug slows Huntington's disease, but is an interesting and satisfying set of relationships that suggest that the plan we already had to do the next trial is the correct plan. But this is basically what we've shown overall. The drug is safe, it is well tolerated, and it produces dose-dependent lowering of mutant huntingtin.
And so it leads to this sort of alphabet soup of things that we have to keep track of to follow this trial. So as we heard this morning, a biotech company like Ionis will develop a drug to a certain point and then often partner with a large pharmaceutical company that has the resources and experience to run the kind of massive study that's going to be required to prove this drug works. And in this case, Ionis has partnered this drug, the same drug, mind you, which they called HTTRX, with Roche and Genentech in the US and Europe. And so now it has the new snappy name RG6042. So remember that. R is for Roche and G is for Genentech, I'm told. And
yes, and 6042 is for 6042.
So remember that. The point is, this is the same drug. It doesn't matter what they call it, right? Each company has a label on it. They have their own labeling system. That's all that's going on here. There's no shenanigans happening. This is just a transfer of, and it's good news that it's happening. And so this is the question everyone wants to ask, right? What's the timeline? Well, the bottom line, as we heard, is we don't exactly know yet. So let's just go through what's happened so far and what we predict and hope will be happening. So the top line results we had as of December, that's great news. The folks in that study, this is really important, were moved into what's called an open label extension. And this gets thrown around, but you might not know what that means. What it means is they take all of the participants who were in the trial and they put them on active drugs. So even the folks that were in the placebo arm of the safety study are now getting regular injections. So that's amazing, because think about it. Although it's a small number of people, because it was a safety study, we can now follow them for a really long time while we're doing the other study in parallel. And who knows, it may not be enough people to see anything, but at least give us hints of what's to come, and also gives us a sense of longer-term safety profiles.
They're also all on the higher doses of the drug, right? So the lower doses didn't produce much lowering. And so in the original trial of 46 people, there were maybe a dozen on placebo, and then another couple of dozen who were on very low doses. So the ones who received those higher doses that gave us that really good news signal, not that many people, but now those 46 are all on those high doses. So suddenly we've got a much bigger group of people receiving the decent doses of drug. And it's not just, so that helps everyone in this room and everyone beyond the room, because it tells us stuff scientifically, but it's also a thank you to them. Mm-hmm. Because some of them, the lady that received my first injection of the drug, that was September 2015. She was finished by Christmas 2015, and then she waited 2 and a bit years for the results to be announced, because that was when we knew that we were going to be doing the open label extension. So this is a huge thank you to them, and they deserve it more than anyone, so definitely good news. Everything gets speculative to the right of the today arrow, so don't hold us to this. This is our best guess.
Yeah. This is what I am personally hopeful to see. So I expect that Ionis and its partners will be putting together a paper describing all the data, not just the safety, so all the little bits and pieces of data. That will come out in the form of a scientific publication. You'll hear about it on HD Buzz. The companies are really good about letting us know and giving us a heads up, so just stay plugged in. You'll hear about it as soon as we do. And then, as we heard from Eric Lundgren today from Roche, there will be a pivotal study. And I mean, I think here today we heard for the first time I've heard publicly stated that the next study will be a so-called pivotal study, a study aimed at getting the drug registered and licensed for Huntington's disease. When is that going to happen? As Eric said today, we're not exactly sure, but it's a lot closer than it was before the safety study was successful.
And so the end of this year is the very earliest I think it could happen. It may well be early next year. And if things turn out to be more complicated, it could be longer than that. But so end of this year is the earliest that trial could start.
And Roche made an announcement today that they're starting a new natural history study they're doing to recruit folks to compare to the people in the open label extension. So it's sort of like a little mini free trial, which is a really brilliant way to maximize the use of those brave volunteers in the first safety study.
So this is one of my slides.
Yeah.
We get about 7 or 8 new referrals a month to our Huntington's disease clinic in London. And in the 2 weeks between the trial announcement on the 11th of December and Christmas, we got 45 new referrals. And then we got 36 new referrals in January. And so we've been seeing a lot of patients in clinic, and they want to know what's happening. They want to know what the trial results mean, what's happening next, can they be in the trial? And it's a totally understandable impulse. And halfway through one of these conversations, it occurred to me that where we are now is exactly like the film Predator. And I'll just leave it there. No. Allow me to explain. So in the film Predator, which many of you won't have seen because you're all so young, there's this really scary bad guy, and actually you can see him here, but most of the time he's invisible, and everyone that comes into contact with him dies, and it's terrifying. And then there's this band of large burly men, including the former governor of California, and some women actually, if I remember rightly, and their job, they are the people being killed one by one by this enemy, and they decide that they're going to turn around and fight. And they shoot all their guns, they see this enemy, and they shoot their guns into the jungle, and they're firing and firing and firing and firing and firing, and the enemy escapes, and they think, oh no, we've wasted all of our bullets. But then Arnie looks down, and there's this blood it's a fluorescent green blood on a leaf, and he looks at it, and he turns to the camera, and in a pivotal moment in the movie, he says, "If it bleeds, we can kill it." And that, ladies and gentlemen, is what we just did to Huntington's disease. So it's important to note that at this point, he doesn't know that this is a fact. This is a belief that he has developed on the basis of physical evidence that he has seen. And he's gone from a position of complete hopelessness into one where he believes that a victory is possible and that a small proof that a small victory can be won leads to him believing that we can win this greater war.
Annoyingly, it actually turned out to be a great analogy.
I know, right?
I was pretty sure it was not going to go that way.
My favorite thing was I gave a brief talk last week in Turin in Italy, and there was simultaneous translation. And before I showed the slide, I said to the translators, "You have to do the voice." And they came up to me afterwards, ran up to me, "We did the voice." I love those guys. Okay, so every cloud has a silver lining. Every silver lining has a cloud. And so I think it's worth stepping back for a moment and saying, what if it doesn't work? We're going to do everything to make sure that we can. But what if something happens? What if we get disappointing news? Maybe we run the whole trial and it doesn't do what we hope it will do. It doesn't slow progression in the patients we test it in. Or what if we get a year into the trial and it turns out that there's some unexpected thing going on that is a problem? These things can happen in clinical trials. We've been really lucky so far. Every time that we've put this drug to the test, it's done really well. It helped the mice, it was safe in people, it lowered Huntington. Everything's been going really well, and I hope it will continue to go well, and everyone's going to work really hard to try and make that happen. But what if it doesn't? Well, I've been saying for ages, a negative trial is not a failed trial. If the trial is run well and run properly, then even a negative result gives us really useful information. So here's my answer, and this was a question that was asked in the Youth Alliance session 2 days ago, whatever today is, subtract 2 days. A bit jet lagged. So the answer is we figure it out. We figure out why it went wrong. Was it a dosing problem? Too high, too low? Doses too close together, doses too far apart? Maybe we chose the wrong group of people. Unlikely, but maybe we chose the wrong group of people and we have to try earlier or later, or we have to run the trial for longer or use different outcome measures. Maybe the drug is working, but we're failing to detect it because we evaluated things the wrong way. The clever people that brought us to this point aren't just going to throw their hands up and go, well, that didn't work, back to square one. Again, if it happens, we'll figure out why and we'll make it work. We'll make it work with this drug, with a different trial design, or we'll change the drug. We'll choose a different drug. We'll use a different approach. We'll deliver it in a different way. Nothing is going to stop us trying. We are going to get there. And so this is the second of my slightly contrived analogies. Again, it came to me while I was talking to a patient in clinic.
For the young people, that's a floppy disk.
No, that's a 3D printout of a save icon. So if you play computer games, whether you're saving it to a floppy disk or to the cloud, whatever that is, you'll know that there are save points. Some games have save points where you have to achieve a certain amount of stuff and then save the game, and you do some more stuff, and if you die before you save it, then you have to go back to that save point. And I think, until recently, our save point was the day the Huntington gene was announced, March 24th, 1993, and that's brilliant, because the start of the game was 1872, with George Huntington publishing the paper, so that was a significant advance. Honestly, I don't think I'm going to be proven wrong. I think we have a new save point. And it's this graph. We did this. Whatever happens next, we know that we can lower the concentration of mutant huntingtin, which is the cause of Huntington's disease, and we can do it in a dose-dependent way, and we can do it safely. And everything else is turning that into clinical benefit. But if something goes wrong in the next phase, we can come back to this save point and say, okay, how do we go from that? What do we change to make it happen and to have success? So I think that's our new save point. And we showed this earlier. It's tempting to look at the handsome neurologist with bad hair or the syringe, but I want to look at this, which is the love handle, and I have written permission to call it that. In fact, I said it by accident at a patient meeting, and the lady was in the room, and she came up to me afterwards and said, I loved that you called it my love handle. That's what it is. I want you to call it that every time you show this photo from now on. And she wrote it down for me. So this is the love handle of the first participant in London. And this is, on the left, is Peter, who I've known for 13 years. This is Peter's sister and Peter's brother. Peter has symptoms, and he was one of the patients in the trial. And his brother and sister both carry the mutation but don't have symptoms. And Frank is a firefighter, and he has 3 daughters. And when we told Peter that we might have a slot for him in the trial, he burst into tears of joy, not because he thought that he would be helped personally, but because he never thought he'd be given the opportunity to help his brother and sister and his nieces. And that is the spirit of the Huntington's disease community that has got us to this point. It's the same spirit that we see in everyone who's ever given a blood sample or had an MRI scan or taken part in Enroll-HD or given a lumbar puncture or completed a cognitive test. And that spirit of generosity to the human race is what is the absolute core of what I hope will be our victory. Please applaud everyone who's ever done anything like that. So George gave you 4 pieces of advice. We've condensed it down to 3, but it's much the same thing.
So people ask me, because I'm in this situation now, that I have access to all this information. I know these folks, although they do manage to keep secrets from me, as has been proven, which was amazing, again. But since when I meet family members, they often say to me, okay, you're a mutation carrier, like, what do you do? What's the secret, that you have access to, because you know all these great scientists? And the unfortunate truth is that right now we don't have treatments, and the best thing that we can do while we're all waiting, and I find myself in this stage where as I age and I have the HD mutation, I notice certain things that change, and I know you all know what I'm talking about, like, did I forget that person's phone number because of HD, or did I do it because I'm getting older? Did I trip on that stair because a cat is I almost said a bad word. Or is it because, my neurons in my brain aren't working perfectly? And the answer for all of us in this situation is probably both. And it's going to be impossible at any time to say, like, oh, that little symptom or thing was because you have the HD mutation and this one's because you're aging. And I have the great privilege and honor that I get to sit around and drink wine with my neurologist friends and play 20 questions with them about it. But in the answer that they give me is take care of yourself. While we're waiting for me and all of you to get access to this drug, the best thing we can do is be healthy and happy and have intact, supportive families when we get there. And that means take care of yourself in all the boring ways. Don't drink too much, eat well.
But also medically, one of the reasons why we can't get people into the trial that we thought would be suitable is that things have changed too much for them in the past few months. Making a trial like this relies on having as little variability as possible within people and between people. So we want, if someone's condition is really unstable going into the trial, if their medications are changing or if their mood is up and down, and no one's going to pretend that Huntington's disease is a predictable disease. But there may be rules like if you've been started on a particular new medication in the past 3 months, you have to wait before you can be in the trial. So if that's going to happen, now is a good time for it to happen, because the trial's not going to happen in the next 3 months. So my advice would be keep up with your appointments with your physician, go to your center of excellence, go to your clinic, keep up, if you're feeling low, tell someone, speak to a psychiatrist, speak to a counselor, speak to a social worker, get your family network in place, and try and get yourself as well as you can with help.
Sign up for Enroll-HD. I know you hear this all the time. Enroll-HD is the best way we have as a community to continue to support the investigators working on HD, both to make basic discoveries about how HD works, but also to recruit for clinical trials.
We messed up the order. There's a thing we need to show you. It's an example of looking after yourself.
Oh, yeah.
Jeff wanted to put this in. I wasn't so keen.
Eating well is an important part of staying healthy. So Dr. Wild's... Okay, I have to set this up a little bit. Dr. Wild's partner is a vegetarian. He doesn't get to eat very much meat at home. So when he travels, you know those like big steaks for two they sell at restaurants? So when we got to Los Angeles, we polished one of those off, and
I'm thrifty.
he couldn't let it go to waste. So
There's meat on the bone. You can't, like, it's an insult to the cow if you don't eat.
The other part of this that's important is this was actually a very posh restaurant, like very fancy people
They
there.
don't want you to waste the bone meat.
So anyway, eat well, take care of yourself. Don't be like Ed. That's the point. The other really important thing: sign up for Enroll HD. Now I'm gonna out myself again here because there hasn't been enough awkwardness today. I was in Predict HD for more than 10 years. I have done a lot of research participation. I have not signed up for Enroll HD.
I'm in Enroll HD. How can you?
Wait, is Dr. Levitt here? He left. Okay, can I get an appointment to come sign up for Enroll HD in Vancouver? All right, there we go. So I am taking myself up to Vancouver to register and enroll.
2 years ago, the title of our talk was The Best Time to Plant a Tree. Chinese proverb, the best time to plant a tree is 20 years ago. The second best time is today.
Here we are.
Living the proverb, Jeffrey Carroll.
And finally, stay informed, right? And so folks want to know, like, when's this trial going to happen? What's the next advance? The best way to do this is to be plugged into research. And I think one thing that's been a really radical change that I think the HDSA deserves a lot, and particularly George Yorling deserves a lot of credit for, is launching of this HD trial finder on the HDSA website. So could we give George a round of applause, please? When George first started at the HDSA, it was great. There was a scientist there, a guy really plugged into HD, and I think it was obvious maybe to him then that it was very frustrating for folks. There's not one place to go to find out about new trials. And so this HD trial finder, you go in, you put in your status and your zip code, and you find out trials that are close to you, and it's going to stay updated, and it's excellent.
Our other recommendation is to follow HD Buzz. It's a little website you may have heard of, founded 9 years ago by Jeff and Ed, and research news in plain language, 14 languages. I won't go into the details, but we get around about 500,000 hits a month to HD Buzz. So we know that the letters that we get, emails, who gets letters? The emails that we get are telling us, and the feedback we get from you guys when we see you in person is that this continues to be a need, and as long as you guys need it, we're going to keep doing it, however much it kills us. So I'm looking into the audience because we have an announcement to make. So there was a guy at our NYA session called Antonio. Is he here?
Yeah, right here.
Where are you? Okay, you're going to meet me at the back of the room.
I'll do a little dance.
So Antonio, we did this Q&A for the NYA on, I guess, Thursday. And basically, it's the Ask Us Anything, but it's for the young people. And the questions are always phenomenal, and they get better every year. And Antonio it's his first time at convention, and he... he doesn't know we're doing this. But he asked a ton of amazing questions. Like, he knows more about stem cells than most of my colleagues. And he was asking about hallucinogenic mushrooms and all kinds of other things. So we're launching today the next update to the HD Buzz website. We're calling it HD Buzz 3.0. It's essentially a new design, a new logo, and a new friendly interface to get all of the news that you love, but in a more kind of pretty way. So what I'm going to do is switch to Safari. It should be loaded up, and I'm going to ask Antonio, aka Question Boy.
Question Man, we decided.
We decided we would upgrade him to Question Man. So to count down with us, 5, 4, 3, 2, 1, and then press the go button. You have to escape the PowerPoint. Oh, we have to leave PowerPoint before it will work.
There's almost no chance this is going to go horribly wrong, right?
We can move that to the other monitor. We'll get there. Like I said.
Fun story about Ed's background. It updates to wherever he is, so you're looking down on us right now.
Okay.
Oh.
Here we are. So HDBuzz 3.0, nothing could possibly go wrong. Count with me, you're just going to press the trackpad. 5, 4, 3, 2, 1, go. Press it again.
Uh-oh.
Hey, it worked. Well done. It's happening live. Congratulations.
Thank you.
Thank you. Okay, you can sit down again. Okay, over to you, Jeff. Oh, can we go back into the PowerPoint? Thanks.
We slightly underestimated how awkward it was going to be for you to walk back and forth across the room.
Yeah, right. Okay.
This is where we should have some witty banter planned.
That's done. Right. Oh, okay.
Good jump.
I don't know why this slide is here, but this should have been earlier. This is another kind of Debbie Downer slide. Okay. So everyone that I've met since December wants to be in the next trial of this huntingtin-lowering drug. Most people who want to be in it won't be able to be in it. However big it's going to be much, much, much smaller than the population of people who have Huntington's disease and want to be in the trial. We've heard today that it's going to be an early manifest HD, which means if you have the mutation but don't have symptoms, you won't be able to be in the trial. And there'll be some people for whom the disease is too advanced for them to be in the current trial. And that probably sounds unfair. There'll be an upper age limit and a lower age limit. There'll be certain medications that you can't be on if you're going to be in the trial, and so on. And so none of that is a failure or by design. The enthusiasm to be in trials is 99% of what matters. And the important thing to remember if you find yourself in that disappointing situation is you did your best. You found out about the trial and you asked about it and you tried to be in it and it wasn't right for you. But there'll be another trial, there'll be another research project, there'll be another thing you can do. And maybe you can help someone else to be in the trial, maybe you can tell a family member that they might be suitable to be in the trial. Fundamentally, the trial is not a means of giving this drug to people for their individual benefit. Right? And some people in this trial, as we heard from Eric, will be on a placebo injection, probably for the full duration of the study, which is going to be a long study. And that might put some people off, and I think that's fine. Okay? So I think the important thing is to have a sincere discussion with your caregivers and with your doctors and team about whether this is the right trial for you. If it is, go for it. If it's not, other people will test the drug for you. Okay? This drug is going to get tested, whatever happens. And the purpose of the trial is to test the drug for the benefit of the human race and for the HD community. So you will still be disappointed probably if you really want to be in the trial and can't, but try and bear that in mind that the drug gets tested anyway and you did your bit. What's next? Yes?
So there's a whole host of other approaches to this Huntingtin lowering thing. And we're talking a lot about the Ionis drug right now because they were the first in the clinic after a long path. And we're all really excited about that. But there are a whole bunch of other approaches. And they come in a kind of a zoo of technologies. But they all have the same idea of somehow interfering with the production of the genetic information and translating it into the Huntingtin protein. So there's things called zinc fingers from Shire and Sangamo. There's gene therapies from Unicure and Voyager and other companies that have folks here that are going to consider other ways of basically achieving the same effect. There's another ASO set of trials that are happening, actually 2 trials run by a company called Wave Life Sciences that are happening now. And these studies are designed at so-called allele-specific silencing of Huntingtin, which means, remember, in the vast majority of HD folks, they have one mutant and one, I'm not supposed to say normal, but since I have one, I feel like I can say normal. One mutant and one normal copy of the HD gene.
One fancy and one regular.
Fancy and regular, yeah, thank you, Nancy. So the idea with this approach is to silence just the mutant version of the HD gene. Now, obviously, that sounds better, and it's like, in theory, the best way to do it is just knock down the mutant one. What's the limitation? Well, the limitation is that these drugs rely on little spelling changes, little variations in the genetic code outside the CAG expansion, the Huntington gene. So you not only have to have the CAG expansion, you have to have inherited these little genetic variations that are the target of these wave drugs. And so not every person can be in these trials. So the benefit of the Ionis and Roche approach is that it will work for every HD patient. And the benefit of the approach being taken by Wave in these two currently ongoing safety studies is that it's a more precise reduction of just the mutant huntingtin protein. Which of these is the better way to go? We don't know. That's why we have trials. And it's amazing that we went from nothing happening for HD to like multiple companies doing really smart stuff for very good reasons, very carefully, to get at the answer to the question, which is which of these is the most efficient way to treat HD.
And way back before any of us really knew about any of the companies that might get involved in these trials, we used to have these theoretical discussions at scientific meetings. Should we do total huntingtin lowering, or should we try and do the mutant selective, the selective form of lowering? There were pros and cons on both sides, and at the end of those discussions, we always agreed we should try both, just in case. And that's what's happening. So, yeah.
And there's other cool stuff. So folks want to know about new technologies like CRISPR or other techniques that allow us to actually, for the first time ever, actually manipulate the DNA of living organisms, including, in theory, humans. And so these approaches are really cool. They're new. There have been 2 studies in the last year in mouse models of Huntington's disease using CRISPR in the brain. Now, this relies on gene therapy. It's much more complicated to deliver than the ASOs, which are simply injected intrathecally. So this is like the next thing, right? So if these ASOs don't work, there's this whole other approach coming behind it that's being tested really rigorously in labs around the world right now.
Right, and this new stuff doesn't stop just because we had one promising result from one trial. Everything happens in parallel. Science is cumulative, to quote George quoting me.
Well, yeah.
It never stops.
And one thing I want to say to people is like, you stop hearing about other things for a little while because the oxygen gets sucked out of the room by the newest press release, right? And that's great because we're excited about something, but don't think that means that everything else has stopped, because it hasn't.
And we're going to give you 3 examples of cool things that have happened in the past year, which are going to be super important, we think, and are going to propel HD research forward. And the first is to do with the huntingtin protein again. A while back, this was the best picture of mutant huntingtin. This is an atomic force microscope, so it's kind of like a little lever that scrapes itself over cells, and however much the lever moves, it produces this picture of a mountain of huntingtin protein. And then a few years after that, using a powerful X-ray, we were able to obtain this funky potato thing, which most of us use in our talks to represent the huntingtin protein. I think that was, 2015 that one was published? About 3 months ago, the problem, this is fuzzy, right? This is not what the protein looks like. It's similar to this, but this is a really kind of fuzzy, cloudy potato thing. The goal of finding out what proteins look like is to identify what's known as the crystal structure of a protein, which is basically an incredibly detailed picture of all of the angles between all of the bits that make up the protein, and then a big picture of how that all folds together and produces the final shape of the protein. It was done. That was how they discovered the structure of DNA. They used X-rays to produce what's known as the crystal structure of DNA. And from that, you can figure out exactly what the protein looks like. And this has been a huge problem, unsolved for 25 years. But 3 months ago, the crystal structure of huntingtin protein was solved. And this is what it looks like. It's huge. It's really pretty. It looks like a load of ribbons, but actually, if you zoom in on those ribbons, we now know the position of every single atom in the huntingtin protein. One of the things that's intriguing us is this: it has a hole, like a pipe going through it, and no one knows what that's for.
Ed asked the scientist the first, this beautiful structure's out, and Ed asked the guy, "What goes in the hole?" He said, "I don't know."
Force of habit. No. Rewind. Computer.
Just keep going.
So this was a team of that was his answer. This was a team of German researchers, and what they did is they froze it they froze a sample of they stuck another protein on the protein to stabilize it like a tent pole, and then they froze it to near absolute zero, and then they fired billions and billions of X-rays at it, and this was the structure that emerged. It's an incredibly big breakthrough, and it happened just 3 months after the news from the huntingtin-lowering trial. Now that we have this, the people who make drugs can go to town trying to find places on this protein where they could stick a drug and interfere with its function. And the people whose job is to study how the protein works and how it's altered in the disease can look at this and say, hang on, I recognize that shape. That's a hinge. That's a face. That's an elbow. That could be a hammer. That's a pair of scissors.
This is clearly not
whatever.
your area of expertise.
This is not You get the idea. You can look the enemy in the face and figure out how it works. This is a big breakthrough, and you'll be hearing of lots of developments that are empowered by this discovery in the coming years.
And there's also been the fruits of all of our efforts as research participants. So we have enormous observational studies. So observational studies are studies that look at HD folks without giving them any drug, right, to learn about how the drug progresses. And so studies like Predict HD and Registry from the European Huntington Disease Network combined, and some researchers led by Jim Gusella and other folks at Mass General formed the GEM HD Consortium. And the idea with this study was to look for genetic modifiers. So we've known for a long time that in certain families, HD tends to occur earlier than you would expect based on the CAG size. And so that kind of, as Jim said, it all starts with families, and that observation led people to the understanding that there are genetic factors outside of the Huntington gene that contribute a little bit to whether you get HD earlier or later than expected. And using massive numbers of subjects, I think the published study was over 3,000 subjects, these researchers were able to narrow in on specific regions of the genome, right? Different areas in our genetic code that help contribute to the onset of HD. And they found variants that both made HD happen sooner and HD happen later. Intriguingly, they found some of those variations in the same gene, right? And so those genes, now we have a whole other set of tools to look at to say, okay, as Jim said, I think we call this out later, but as Jim says, like, the nature has already done the experiment. So if you're thinking, like, is it possible to change the onset of HD? The answer is yes, because if you have this particular genetic variation in this other gene, you're likely to get HD later, and we know that. So it's a proof of concept, and it's a great target for people who make drugs to say, look, if we can mimic whatever's happening to that gene with a drug, maybe we can have that same effect in other people that don't have the mutation. And I think the latest version, Jim, of the GEM is about 9,000 people. So people who donated their blood years ago, maybe it got used in ways that you never could have predicted at the time.
DNA repair.
And so this was these findings were replicated in another genome-wide association study led by Sarah Tabrisi and colleagues in University College London. And both of these genetic modifier studies came up with the same answer, which is that genes that have to do with the process by which cells repair their DNA, those are the genes where those variations occurred, the variations that led to earlier or later onset of HD. So I think if you asked a bunch of HD scientists, what kind of genes will we find that modulate the onset of HD, DNA repair probably wasn't going to be super high on the list. It's an interesting finding, but really striking.
Right, and it was a common finding across many of the genes. That they will all seem to be focusing on the repair of DNA, which is kind of cool.
This is an awful picture.
We were going to replace it, but then we ran out of time, and he put the video of me in, so...
I look like a prisoner. What am I doing?
You're demonstrating somatic instability.
Yeah. So the hunch, what we think is happening here is this process that these DNA repair genes are influencing is a process called somatic instability. And all that means that if you're like me and you inherited a CAG size of 42 from, in my case, my mom, that's the repeat size that you'll see if you took a blood sample and sequenced it. However, in certain cells of the body, including cells in the brain, if you went and looked in that same individual, instead of having a CAG of 40, you might find CAG of 50. So in certain tissues, it's almost like the DNA slips or stutters as it repairs, and that CAG expansion gets longer. Now, we don't make babies with our brains, so you're not going to
evolved.
Anyway, you're not going to pass on that unstable, say, 50, but it's going to be affecting your brain cells. And so because these genes that modulate onset also interfere with DNA repair, we've become really interested in this process of somatic instability.
And we now have like a ton of new targets and work that can be done based on that to turn that into drugs, as Jeff has said. So yeah, that's cool. Mother Nature's already done the experiment. And as Robert said, there's nothing more precious to a drug hunter than an observation made in the population you want to treat. And that's exactly what this is. And you can expect that to lead to big breakthroughs in the coming years. And the way to make that happen more quickly is to join Enroll HD. It's like we are a stuck record. Our answer to everything is going to be sign up for Enroll HD because it's a study that is designed by scientists with HD family members to help us work together to give us the resources that we need, the blood samples, the gene samples, the clinical data that we need. So please do that.
Oh yeah, the pig. And so just to give you a sense, it's not just that we're only working on ASOs or the next ASO. There's whole new approaches being brought to bear on HD. So there was recently a publication in a huge prominent journal of the first time that people used the CRISPR, this new genome modification technology, to actually edit the DNA, in this case of a pig, a large animal. Now, you might say, like, why use large animals? Because they have big brains, and so if you're testing something like gene therapy or ASOs, it's way nicer to work in an animal that has a big brain like a human rather than mice, as much as I personally love mice. So there was a huge publication by the group of Zhao Li who described this pig. So you can see there's two pigs here, and one of them that's indicated HDKI. You can see its diaphragm flexing, and it's having a little problem walking.
So it has involuntary movements, which is one of the symptoms of HD. And if we play the video again, you'll see that it just has some slight stiffness in one of its back legs.
So this is really important. None of the mouse models of HD do this.
And it's wagging its tail, and I looked it up, and that means the pig is happy.
So what does this do for us? It tells us that researchers, even using the most cutting-edge tools, they could have made any genetic disease model, right? The first time this was ever used was to make an HD model. So I would sometimes feel like in the HD community, we feel like we're unseen, and I want to say it's definitely not the case. These new tools are being used in really cool ways.
So here we are. It's 2018, 25 years since the discovery of the gene, and it's been an incredible year. We have a new save point, and great things are happening, not just in the Ionis-Roche Genentech program, but across Huntingtin-lowering and other therapeutic developments and basic science developments that will seed the next generation of progress in HD. The road ahead looks good, and as we've said, though, there will be challenges, but we can solve them together. And in 2009, Sarah Tabrizi gave the opening, no, 2011, Sarah Tabrizi gave the opening talk at the World Congress of Huntington's disease in Melbourne, and she quoted Winston Churchill. And I made this Photoshop at the time. And what she said was, and people may know this Churchill quote. I'm not going to do the voice. This is not the end. I'm not going to do Sarah's voice or Winston Churchill's voice. This is not the end. It is not even the beginning of the end, but it is perhaps the end of the beginning. I agreed with that in 2011 because that was based on a ton of stuff that we'd discovered in the lab and through work on patients. I think in 2018, we may have just seen the beginning of the end. So, which wolf wins? It's allegedly an old Native American proverb, but it probably isn't. It was probably made up by white folks and then attributed to Native Americans to make it sound more mysterious. It's a good story, though. It's a story about a kid who's talking to his grandfather, and the grandfather's giving him life advice. And the grandfather says, you know, within each of us, there are two wolves, and one wolf, and they're constantly fighting, and one wolf is anger and fear and despair, and the other wolf is hope. And the kid says, which wolf wins? And the grandfather says, the one you feed. So I think that is our take-home message for you guys. Feed the wolf of hope. Be the change you want to see in the world. Look after yourselves. And sign up for Enroll HD, and let's make this happen together. Jeff wants to say something similar, but it comes from Woody Guthrie.
I love this. So if you haven't seen this before, this is Woody Guthrie, who, of course, most notably for us was the husband of Marjorie and the founder of the HDSA, but also like an amazing poet. And he wrote these New Year's resolutions. And there's some notable features. One, that in the top and the middle, it says middle of book. and there's a whole bunch of really sage advice, like wash teeth, if any.
I like 27.
Which?
Help win war, beat fascism.
And he says, my favorite is number 19, which he says, keep hoping machine running. And I think that's the best thing that me and all of us in this community can do, and I encourage you to do it over the next year, and we look forward to seeing you this time next year. Thank you.
Thanks, guys.